Signaling is the general mechanism by which cells respond to their environment and by which multicellular organisms can coordinate the behavior of cells in a tissue so that they work together and they can pattern each other. They can signal each other to specify cell fate in development. We're going to talk about some examples of how signaling works to give you an idea of the types of mechanisms that operate in cell signaling, but it's a vast subject, and any variation on the themes that I'm going to talk about today is probably used somewhere in biology. So this is just an example that's mentioned in the book of single cell organisms responding to a signal. These are budding yeast cells, again, Saccharomyces cerevisiae. And budding yeast cells, they eat and they divide and they can respond to other kinds of signals in their environment. But in this case, they are responding to pheromone signals from other cells. So yeast cells come in two flavors, A and alpha. They're called mating types. Each of the mating types secretes a soluble pheromone that the other cell type responds to. And that's what's happening here. These cells are sensing
chemical signal or pheromone, and in response, they are changing their shape. They are putting out these projections on one side that will enable them to mate with other cells. Within multicellular organisms like animals, signals are often produced by one cell and then sensed by another cell that responds to the signal. When these signals travel through the bloodstream, often for long distances between the cells that make the signal and the cell that receives them, those are called endocrine signals. There are also paracrine signals, which are secreted by cells into the extracellular fluid and sensed by nearby cells, but they don't travel nearly as far as endocrine signals. And then there are two types of close-range signals that are mentioned in the chapter. One are signals secreted by neurons in response to an electrical signal that are then received by a dendrite on an adjacent neuron. And the other are signals that are present on the surface of cells that are bound by an adjacent cell, by receptors on that adjacent cell, in direct contact with the first cell.
So most of today's talk will focus on the first kind of signaling, where you have a small molecule signal that is coming in from outside the cell and somehow being responded to within the cell. There are two classes of signal molecules. The one that I will focus on mostly today remains outside the cell. It can't go through the membrane, and so for this cell to respond to whatever the signal is, requires receptors on the surface of the cell to somehow sense the signal and transduce the information that that signal was sensed into the cell. Another class of signaling molecules, particularly some kinds of steroid hormones, are small and they're hydrophobic enough that they can actually pass through a membrane, and they can directly go into the cell, bind to receptors within the cell, and many of the receptors for these kinds of signals, once they bind to the signal, they go into the nucleus and directly activate transcription. This is the only slide I'm going to show about that kind of signaling. It's relatively simple compared to the rest of what I'll talk about today. But these are, of course, very important signals in development and reproduction. Things like estradiol and testosterone have this sterol-based structure that enables them to pass through membranes. The receptors are there in the cytosol, but once they bind to the signal, then that triggers them to go into the nucleus, and they can either bind directly to DNA or they can interact with other proteins that bind to DNA to activate transcription. The book points out that the number of different signaling molecules that are used by our bodies is not nearly as diverse as the kinds of responses that are elicited by those signaling molecules. And so a given signaling molecule like acetylcholine, which is the structure shown here, will trigger different responses in different cells. And sometimes it's because the receptor is different. So here there's a receptor for acetylcholine that's actually pretty similar, maybe identical, I'm not sure, between a heart pacemaker cell and a salivary gland cell, but the same small molecule signal will bind to a different kind of receptor, in this case, a channel in a skeletal muscle cell. And even if it binds to the same receptor on two different cell types, the downstream components that transduce the signal may be very different. And so the response within the cell will depend on both the receptor and downstream components. So that's one way that organisms can diversify their responses to simple signals. In addition, most animal cells integrate many different signals all the time. So they're getting signals from the bloodstream, hormones, nutritional signals, et cetera. And they have many surface receptors that are receiving these signals and integrating the information to decide whether to grow, whether to store fat or hydrolyze glycogen, depending on the cell type and its possible fates. It will respond to signals in a combinatorial way. And often the complete withdrawal of signals will trigger a cell to undergo programmed cell death, also known as apoptosis. And so that is just illustrated here. The cell isn't getting any signal, so it dies. The response time to a signal can vary from microseconds in the case of neurons to many hours or days. And in part, that depends on what the response is. I talked about the case of steroid hormones that go into the nucleus and activate gene expression. That is a relatively slow process. Movement of the receptor into the nucleus and binding to DNA can be very fast, like on the scale of seconds. But it takes time to transcribe new RNAs and for those RNAs to then go and get translated into new proteins. And so the cell spends hours often adjusting its physiology in response to signals. Most of the examples we'll talk about today involve faster kinds of switch mechanisms that can happen on the scale of seconds to minute to dramatically alter cellular behavior. And that's often through enzymatic pathways. So they don't have to make anything, they just need to modify existing proteins. As I'll talk about, in order for a signal that stays outside the cell to do stuff inside the cell, there are typically internal signaling mechanisms that are activated in response to this extracellular signal. And so there's a relay of information from outside the cell to inside the cell. And there are enormously complex ways in which this can happen. And I'm just going to touch on a few of the key mechanisms that are shared by many signaling pathways, just to illustrate the variety of different kinds of pathways. And then these are the kinds of outputs that are elicited by these signals downstream of all this internal signaling. You can have, as I said, changes in enzymatic activity. We haven't really talked about the cytoskeleton yet, but if a signal is telling a cell to, for example, move towards food or away from some adverse signal, that will often lead to changes in the cytoskeleton. And we've already touched upon the fact that transcriptional outputs or changes in gene expression are a frequent response to extracellular signals. The chapter groups these receptors that respond to extracellular signals into three main classes. And I'm not going to really talk about these because we haven't really talked very much about ion transport across membranes. If you're interested in neuroscience, that's obviously a super important mechanism of signaling, but it's also important in many other cell types where an extracellular signal will open channels in the membrane and allow ions to come in. And then the ions act as sort of a second messenger to induce changes within the cell. I will talk mostly about G protein coupled receptors, which are a vast family of receptors. I think there's something like 700 in the human genome, not counting the olfactory receptors, which are the receptors in your olfactory epithelium in your nose that respond to smells, of which there are over 1,000, and those are also G protein coupled receptors, each of them tuned to slightly different odorants. Later in the talk, I'll just mention another class of receptors that are actually enzymes. Distinction between these two classes is that G protein coupled receptors execute the key distinction between these two classes of receptors is that G protein coupled receptors, as you might imagine from their name, execute their effects by interacting with these G proteins. Enzyme coupled receptors, as I'll talk about, also can interact with and mediate signaling through G proteins, but they're a little bit different, and we'll talk about how. G protein coupled receptors interact with what we call heterotrimeric G proteins, so I'll show you how those work. As I said, this is all I'm going to say about these channels that in response to an extracellular signal open and allow ions to enter the cell. These are also called transmitter-gated ion channels, and I decided that this is something I don't know much about. You're welcome to read more about it, but I decided to focus on the other kinds of receptors for today's lecture. In order to understand how receptors activate internal signals within cells, you have to remember that there are a number of different kinds of proteins that can act in a switch-like fashion. And we already talked extensively about a kind of G protein or GTPase, which can behave as a molecular switch. And that was RAN, which I talked about on Monday, I think, which is involved in nucleocytoplasmic transport. I'll talk about these more elaborate G proteins today, but just to remind you, G proteins or GTPases exist in two states. They exist in an on state where they're bound to GTP and an off state where they're bound to GDP. Often G proteins are involved in intracellular signaling by switching between these two states in response to an external signal received by a receptor. The on or off state of these G proteins then contributes to downstream signaling within the cell. Another major class of internal enzymes that act as switches are kinases. Kinases are enzymes that transfer phosphate from ATP to other proteins. And the phosphates on the surface of those substrate proteins then modify their activities or their interaction with other proteins. The activity of protein kinase is countered by protein phosphatases that remove these phosphates and change the activity of the targets, turn them off, or sometimes it's the reverse, that the kinase will turn something off and the phosphatase will turn it on.
Someone has a question. Is the on state being GTP bound, is that always true, or can you have an on state?
I think it is not 100% of the time, but I don't know of a specific example where the GDP bound state is the on state for a G protein, but I think they exist. So we talked about RAN, as I said, on Monday, and this is just to remind you that RAN itself is a GTPase, so it binds to GTP and it hydrolyzes it to GDP, but by itself, it does that pretty inefficiently, very slowly. G proteins or GTPases are very often acted on by other proteins, regulatory proteins, that are classified as GEFs or GAPs. GEFs are guanine nucleotide exchange factors, which promote the release of the hydrolyzed GDP and the binding of new GTP. And so they can turn the G protein on if its on state is GTP bound. And conversely, GTPase activating proteins or GAPs stimulate the GTPase activity of G proteins and thereby catalyze them turning from on to off. And that is true for both these small monomeric G proteins like RAN, or I'll talk about RAS in a little bit, and also often true for the trimeric G proteins that work with G protein coupled receptors. Here is a little bit more detail about G protein coupled receptors. So as I said, this is a very large family of receptors that can respond to small molecules or larger molecules in the extracellular space. But this is a family of 7 pass transmembrane proteins. The way these work is that they bind to a signal on the extracellular surface, and that either recruits and or activates a G protein that is bound to the inner side of the plasma membrane.
When the receptor binds to the signal or the ligand, that induces a conformational change that is felt on the inner surface of the receptor and affects its interaction with the G protein.
This is just 2 versions of the same thing. This is the cartoon version of the G protein coupled receptor with the ligand or the signal bound in the extracellular surface. And this is actually a crystal structure showing the 7 transmembrane helices that are common to this whole class of receptors. So they're all evolutionarily related to some ancestral receptor molecule. They're very versatile. They can interact with different ligands, small ligands like this one, or even large ones like peptides or proteins. And within the cell, they interact with different G proteins that are all at the plasma membrane. This gives a little bit more information about how the signal transduction process actually works. Typically what happens is that you have a G protein coupled receptor in the plasma membrane. It senses the presence of a signal and binds to it, and that recruits this heterotrimeric G proteins. By default, the G protein is usually in the off state, so it's bound to GDP. But when it interacts with this receptor that has bound to the signal, that induces a conformational change in this alpha subunit. There's 3 subunits, alpha, beta, and gamma. That's why they're heterotrimeric G proteins, 3 parts, all different. So the alpha subunit is the GTPase. When it interacts with this receptor that's bound to the ligand, the receptor essentially acts as a GEF. It triggers the release of GDP by the alpha subunit and the binding of GTP. By turning on the alpha subunit, that also causes the alpha subunit to dissociate from the beta gamma subunits. And each of these can then activate other proteins. These are all types of what's called allosteric interactions, where something happens on one side of a protein and that triggers something to happen elsewhere on the protein through inducing some kind of conformational change.